Hungry. What are you up to? I just learned everything about sensory systems.
Hello again, Pesky. I'm just about to eat these French fries.
That sounds like an opportunity for me to enlighten you. Would you care for me to tell you how your senses work as you experience those French fries?
Please, no, just let me eat. Leave me alone.
Great! You're in for the ride of your life. There are two types of senses you can consciously perceive. The special senses of vision, hearing, taste, smell, and equilibrium, and the somatic senses of touch, temperature, pain, itch, and proprioception. I'm going to tell you about the special senses because you're special to me.
I barely know you.
All sensory pathways begin with the stimulus acting upon a sensory receptor. If the stimulus is above a certain threshold, the receptor converts it into an intracellular signal, and action potentials move from the sensory neurons into the central nervous system. To be perceived consciously, signals must reach the cerebral cortex. There are all kinds of sensory systems, from ones as simple as a single sensory neuron with branching dendrites to complex multicellular sense organs such as the eye.
I don't care.
Sure you do! There are many different kinds of receptors in the sensory system, including simple neurons with free nerve endings, complex neurons with nerve endings enclosed in connective tissue capsules, and specialized neurons, which release neurotransmitters onto sensory neurons to initiate an action potential. The special senses have highly specialized receptors. Olfactory receptors, smell receptors that is, are neurons, but the other special senses use different types of receptor cells that can send signals to sensory neurons. There are 4 major groups of receptors that are all receptive to different stimuli. Chemoreceptors, such as those of taste and smell, are sensitive to chemical ligands that bind to the receptor. Mechanoreceptors, such as those of hearing, are sensitive to mechanical energy. Thermoreceptors are sensitive to temperature, and photoreceptors, such as those of vision, respond to light. Doesn't that just light up your life?
No.
I know, right? Each sensory receptor has an adequate stimulus, a form of energy to which it is most responsive, although it can often respond to other forms of energy if they are intense enough. The threshold is the minimum stimulus required to activate a receptor. Now, how, you ask, do receptors convert these stimuli into electrical signals?
No, I never asked that.
This process is called transduction. In most receptors, this is accomplished by the opening or closing of ion channels, which causes a change in membrane potential by an influx of cations that depolarizes the membrane. The change in sensory membrane potential is called the receptor potential, in which some cells can initiate an action potential that travels to the central nervous system. In other cells, the receptor potential influences neurotransmitter secretion, which alters electrical activity in a sensory neuron. Isn't that just electrifying?
Pesky, please. As you can see, I'm just trying to eat my French fries.
As I can see. See?
Ah, my eye!
Do you want to know how we can see things? Do ya? Do ya? Of course you do! Your eyes are sensory receptors. They focus light on light-sensitive surfaces called retinas and use a lens and an opening called the pupil. Just like a camera, right? Photoreceptors in the retina transduce light energy into an electrical signal. Neural pathways from the retina to the brain process these signals into visual images. So let's take this French fry here. How can you see this lovely golden stick of wonder?
Can I have my eye back?
Of course I can tell you. Light waves bounce off the fry and are reflected into your eye. These light waves enter the eye through the cornea and pass through the pupil, striking the lens, which focuses the light to hit the retina. There are 2 types of photoreceptors, rods and cones. Rods help you see in the night, when things are in black and white. Cones help with color vision during the daytime, when there's more light. Visual pigments are bound near the photoreceptors, and these are the transducers that convert light energy into a change in membrane potential. The last step in this whole vision thing is signal processing. Photoreceptors converge into bipolar neurons. Ganglion cells are the next step on the neural pathway. Action potentials leave the ganglion cells and travel down the optic nerve to the brain. Now the optic nerve enters the brain at a point called the optic chiasm. Here, nerve fibers from each eye go to the opposite sides of the brain.
I wish we lived on opposite sides of the world.
What? That's not what I said. You're just hearing things. Speaking of hearing things...
No, please stop. Let's talk about hearing.
Whoa, what just happened to your voice? What happened to mine?
That's just how your brain is interpreting the noise our voices make. Let's see how this works. Hearing is a really complex process. Energy from sound waves in the air become mechanical vibrations within the ear when they hit the tympanic membrane, or the eardrum. The vibrations go down the three little bones in the middle ear, the malleus, incus, and stapes, which amplify those vibrations. When the stapes vibrates, it moves the tissue of the oval window, part of the inner ear, which creates waves in the cochlea, the fluid-filled part of the inner ear. The fluid waves open ion channels in hair cells, the sensory receptors for hearing. Ion flow in the hair cells creates electrical signals that in turn release chemical signals by way of neurotransmitters, which starts action potentials in auditory neurons.
Well, this lunch break with you has sure turned my world upside down.
Upside down, huh? Your sense of equilibrium is what helps you determine when your body, or world, is upside down. For equilibrium, we're going to stay in the ear. Equilibrium can tell us about our movement in space and whether our heads are in an upright position or not. Equilibrium is mediated by hair cells that line the vestibular apparatus, which is in the inner ear, like the cochlea. These hair cells are like the ones in the cochlea, except that they're influenced by gravity and acceleration rather than sound waves. Endolymph fluids fills the vestibular apparatus, and when you turn your head one way, it turns the hair cells in the opposite direction, which gives the sense of rotation. When you move your whole body or even just head position, the otolith organs are able to sense linear force, and the hair cells are activated based on movement. Based on these patterns of hyperpolarization and depolarization of the hair cells, your brain can calculate your head's position in any body movement.
Please go away. My fries are getting cold. This stinks.
Stinks? It smells fine to me. The sense of smell is a form of chemoreception, one of the first senses evolved by living organisms. The human olfactory system is made up of primary sensory neurons forming the olfactory nerve, which is connected to secondary neurons in the olfactory bulb of the brain, where complex processing takes place before signals pass through higher-order neurons to the olfactory cortex. Human olfactory neurons are concentrated in a small patch of specialized epithelial tissue in the nasal cavity. These neurons have one dendrite extending from the cell body to the epithelial surface, and one axon extending to the olfactory bulb. These cells have very short lives and are constantly being replaced by stem cells. The surface of the olfactory epithelium is covered with the terminals of these olfactory sensory neurons, knobs covered in cilia that function as dendrites. There's a layer of mucus over this that odorant molecules must dissolve in before binding to an odorant receptor protein. The binding activates the opening of ion channels, depolarizing the cell and triggering a signal.
Leave. I'm hungry. I just want to taste these fries.
Hi, Hungry. I'm Pesky.
Ugh.
The sense of taste, gustation, is closely related to the sense of smell, olfaction. Gustation is also a form of chemoreception. Although there are hundreds of types of olfactory neurons, taste is believed to only have 5 types: sweet, sour, salty, bitter, and umami. Taste receptors are primarily located on the taste buds on the surface of the tongue, but they are also scattered throughout other parts of the oral cavity.
No!
Much like how the sense of smell works, for substance to be tasted, it must dissolve in the saliva and mucus and then interact with a membrane protein, initiating signal transduction. In sweet, umami, and bitter taste cells, receptor proteins are associated with the protein gusductin, which activates many signal transduction pathways. Salty and sour transduction, in contrast, appears to only be mediated by ion channels, where an influx of hydrogen or sodium cations depolarize the cell and send a signal that causes you to interpret the tastes as sour or salty, respectively. And in conclusion, those are your special senses.
You're finally done? Sweet fries.
Hey, Hungry.
Yes.
a french fry?
No!